this is what brought me into the space initially, is the hope that, we can get beyond insulin treatment, but move into curing people instead. Why do we have to inject every single day? Why do we have to be worried? Why do we have to have these complications, all the problems associated, say, living with diabetes for a lifetime, when the cure is so obvious? Just put new cells in. It has been the thing that has always driven me, the simplicity of addressing that is there, but the complexity of making the cell has always been the thing that has followed.
Let's discover the Cleveland entrepreneurial ecosystem. We are telling the stories of its entrepreneurs and those supporting them. Welcome to the Lay of the Land podcast, where we are exploring what people are building in Cleveland. I am your host, Jeffrey Stern, and today I had the real pleasure of speaking with Jensen, PhD, CEO, and founder of Trailhead Biosystems, based here in Cleveland. Jan is the lead inventor of Trailhead technology and has 20 years as a molecular developmental biologist. He is the Eddie J. Brandon Endowed Chair of Diabetes Research at the Cleveland Clinic and obtained his PhD from the University of Copenhagen in 1998 and has been a faculty at U.S. institutions since 2001, where he has published more than 50 peer-reviewed papers and is now engaged with multiple research projects and consortia covering neural, renal, pancreatic areas, as well as cancer and immunotherapy. The technology of Trailhead Biosystems, which exists around the high-dimensional design of experiments dependent on computerized designs and robotic executions, allows for the production of cellular ink as the biological building blocks to address a myriad of issues across cell-based therapeutics, drug discovery, organ printing, and disease modeling, ultimately aiming to arrive at the cures for diseases we've historically only been able to treat, like diabetes, which is one of Jan's original motivations. Trailhead Biosystems is one of the most fascinating companies I've come across so far, and I have learned so much from this conversation with Jan. Everything from the basics of cell differentiation and developmental biology to the vast implications that unlocking industrialized manufacturing of specialized human cells can have for regenerative medicine. So please enjoy my conversation with Jan Jensen. So more and more these days, I feel like I am hearing about biology, developmental biology, synthetic biology as kind of the next great frontier and where we can realize, some of the greatest gains for human development going forward. And with that, I am excited to hear about all the work you're doing in the space. And I want to start maybe with just how you found yourself in the space. Where did you come to develop your passion for biology?
Oh, yeah, thank you, Jeffrey. I mean, it wasn't planned at all. It was my career has more or less been, if the group went in one direction, I decided to go in the other because we couldn't all go in this particular direction. And it's been like that ever since I went through high school in Denmark, where my class wanted to become engineers. And while I was interested in it, I couldn't feel like, we all should do that. So I had to do something else. Personally, I don't have a background in my family at all within sophisticated research. Nobody in my family had an advanced degree. I didn't know what that would entail, but the doors opened. I come from Denmark, and it's really an advanced society today, but I'm still the first generation from a family that sort of came out of the farming generation. And the doors opened for me because of the free education, and the system was there. And I was a kid that just enjoyed sort of mathematics and physics, but never even thought about daring to jump into something as crazy as science. So coming from a military family too, my father was in the military for his entire life. And I wanted to be more or less a fighter pilot, but I couldn't because of my eyesight. I ended up eventually within the University of Copenhagen. And that was back in the early 90s. And then I somehow realized that, because I decided on biochemistry. And biochemistry was because it was not engineering, and maybe I could do research if I became a biochemist, but I really had no idea. So I threw myself at biochemistry and realized then that the more effort I put into this, the better I became. So it all, one step took another. I went from biochemistry, I went into molecular biology. And then after a couple of years in that, I then realized that this thing called cell differentiation, that's what my heart was actually beating for, because it was so complicated. And we didn't really know what controlled it. At that time, we had just begun to find the first genes. There were some fantastic papers, and there were some distinguished scientists in the United States. And I saw a presentation by Bob Tejan from Berkeley. Came to Copenhagen, gave a presentation. I was blown away of how sophisticated that type of science was. So I was more or less just, this is what it's got to be. So I threw myself and attempted to become a researcher and did a PhD focusing on these transcription factors that are controlling genes. And I was fortunate to actually do this in a very large company, in Novo Nordisk. While I was still getting the degree at University of Copenhagen, I could still do it at this fantastically endowed research institute that was owned by Novo Nordisk. It was called the Hagedorn Research Institute. And with that name actually came one of the renowned diabetes research institutes in the world. And I did not know really what it was I was finding myself in. But by happenstance, my interest in cell differentiation landed me right in the middle of the field of diabetes. So that's where it started.
Wow.
And since then, I've just been moving through life as a scientist, mainly focused on curing diabetes, which is also part of the reason that Trailhead exists, which we can talk about. But yes, it sort of became a happenstance role of an emerging scientist that just wanted to see what was behind that door that I didn't even know existed.
Oh, that's amazing. I feel like I'm going to have to ask a bunch of clarifying questions given the nature of the topic that we're discussing here and the complexity of it. But just to kind of set a baseline, what is cell differentiation?
Yeah, so if we look at our cells, we consist of cells. Most people know that. And there's a couple of trillion cells in our bodies, but there's not that many different types. Altogether, we have around 500 or so different cell types. So for instance, if you're looking at your liver, the cell type that's most prevalent there is called the hepatocyte. That's the cell that does most of the workload of the liver. But there are some other cells too. There are some that are called cholangiocytes, and they're the ones that collect the bile into the gallbladder. And you have got endothelial cells that provide the blood vessels for the liver too. And there are more cells. So without going into detail with how many cells that may exist in the liver, each organ consists of a different set of types of cells. Now, cell differentiation is essentially the process by which cells become different. They're in the name. And then what the heck does that mean? I mean, and now you have to think about where we actually come from. Our very first existence happened at fertilization. Thank our mother and father for that event. But after that, we're sort of a little bit on our own, initially, of course, in the uterus. And it's a beautiful thing, this thing called life, actually, from a biological standpoint, also from everything else. But it's pretty fascinating what happens after fertilization. Because that cell, that oocyte that now got fertilized, which now consists or has all the biological information that it needs encoded in the genome to create something as complex as we are as we speak here today, is essentially, that's where cell differentiation comes in and is essentially explaining how all of these cells became different and also numerous. So in the embryo, which we now refer to it, I mean, is a process of cell proliferation, and then sometimes cell specialization. And the specialization is what we call differentiation. So all of a sudden, there is a change inside that growing embryo that is now not homogeneous anymore, but it begins to, from the outside, actually we can see patterns emerging. We can see that there are ridges coming where the brain would be. And it emerges into the process of what's called gastrulation, which is the first true visible evidence that there's something really complex being formed here, our organism. And when that process happens, this is like an orchestra playing all at the same time. It's beautiful. All that information that's been packaged into the genome over the millions of years that has led to where we are today as a species is now being interpreted. And it's being interpreted in space and in time. So as the embryo grows, it gets larger and larger because there are more and more cells. And the patterns, and in developmental biology, we refer to it as patterns, because we can see these changes in gene expression and emerging structures of the embryo that is now being created. This is governed entirely by the genome and how the genome is being interpreted. So if, for instance, one field of cells becomes specialized or differentiated, it will now begin to signal to others so that they also can become differentiated. And therefore, it's actually a process where the cells in the embryo communicate with each other all the time as they help each other to specialize. And in developmental biology, which is a field that I learned, we call this process, is sophisticated, we call it vertebrate regulative development. And it means that the cells are communicating. There's not a single cell in the embryo that is given its ultimate destination from the outset. They can all, in a very plastic way, communicate and then build the robustness up of that eventually creates us as an organism. One example I can give you is, for instance, our developing limbs, say the arms.
Sure.
They look very similar. If you look at the right, if you look at the left, it's like the mirror images of each other. All that information that was encoded to build them was in the genome that was interpreted in two different fields, one on the left side and one on the right side of the embryo. And they never communicated to each other. So the left arm, the information there was not traveling over to the other to say, you should be a little longer or something like that. No, the process here is entirely interpreted within the field from which they arose. So it just speaks about how strict the rules are followed in the normal embryo. And I think as a biologist, it's like, this is where I find comfort. I find comfort in the strength of biology because it works really, really well if you're not messing it up by putting it into sort of an artifactual environment as a cell culture incubator is or a bioreactor. This is not the normal environment, and therefore the cells behave very differently. And that's one of the challenges we have in the field. But the embryo itself, that's the safe harbor. That's where the process of differentiation occurs unperturbed and well.
Fascinating overview. Thank you for that.
There's a little lecture in developmental biology, but few people actually ever hear it. And I think it's a story worth telling because we all went through it.
Right. No, of course. So where does your path and interest in cell differentiation and developmental biology converge with your presence here in Cleveland?
So that's also happenstance. So you see, I as I said, I landed in the pancreatic field because the pancreas is the organ in which the pancreatic insulin producing cells reside. And my first research back in Denmark was actually focused on where do these pancreatic islet cells come from? Because some investigators thought that they came from what's called the neuroectoderm. And I just couldn't come to grips with that as a scientist because when I looked at the embryo, it didn't look like that. So most of my first research was actually trying to sort of help in the understanding of, these cells are from the pancreas. They are formed within the pancreas. And so I was spending most of my early career studying the very, very basic science of this formative process of a cell that produces insulin. Now, those years were in the 90s. And from a scientist perspective, it was quite successful because I managed to publish some publications that then led to, the capability that I could move on to the next stage in my career. And that then was a question that was given to me, strangely enough, by a German that was participating in a conference in a Swiss resort, beautiful Swiss Alps. And he asked me, because he was a professor at University of Pennsylvania, and he said, "Hey, Jan, what do you think about, coming to the United States and becoming a researcher?" I had no idea what that would mean. So I said, "Tell me more about it." "Well, you become an independent researcher, you will get some grants, some support, and you can decide what you want to do." And I felt that was unbelievably fascinating to think about, because I wasn't really geared up for a university career in Denmark, I thought. So I managed to convince my girlfriend, now wife at the time, Sina, was it okay if I was sort of, for a little while, thinking about perhaps going to the United States? She said, "Okay, but only for 5 years. Thank you very much." So back in 2001, I applied at the University of Colorado for a position as a researcher, as a young faculty at that university. And I got the position, and then I moved almost immediately. And then I was at what's called the Barbara Davis Center for Childhood Diabetes, which was a very good, still is, institution in the US focused on diabetes. And so I spent a couple of years there, actually almost 7, to build a laboratory up, and I was focusing on development of the pancreas and the islet cells, so I continued that research. Thank you very much to the American Diabetes Association for providing the early funding for that. But after a couple of years, after having established the lab, I received a letter from here in Cleveland. There was this strange institution I actually did not know too much about, I have to admit. It was called the Cleveland Clinic. The name had sort of, I heard about it before.
Sure,
but
small system.
Yeah, well, it was mainly because I didn't really know much about diabetes research in Cleveland, and there was not too much going on. And now, of course, there was a good amount of maybe clinical research, but basic research was not too much. So there was an interest in seeing if that could be started up here, and I got convinced, that was a good idea. So I moved to Cleveland and became staff at Cleveland Clinic. And I was there for a total of, 13 years before I moved here to Trailhead. And therefore, I could continue the laboratory and perform studies on the same subject matter, which is how does the pancreatic beta cell develop and can we cure diabetes? So that's what took me to Cleveland. And the 5 years that my wife said we could be in the US quickly turned into 22 and going. And now it's Cleveland, and this is the place we call home.
So how then do you bridge this gap from research to entrepreneurship? And as you think about, Trailhead Biosystems and the work you're doing now, what was the founding story there? What were the questions you were asking? What did you have a vision for the kind of work you would be doing now?
Yeah, I think the vision has always been there, but to convert it into reality in research means, how do you find funds to do that? And that's hard in the academic world. It's very, very challenging, actually. And that's just how the system is built in general. Back in 2011, the idea to create a company here in Cleveland based on the technology that we were developing at the clinic was fostering the thoughts about, let's see if we can build a company around this and then add another technology that would then be contributed by another company actually outside the country. So there was a good amount of effort in that phase between 2011 to 12 in attempting to actually create a diabetes-oriented company addressing the disease curatively. But it never actually came to fruition. And after that happened, of course, there was a bit of frustration because there's a lot of effort that was put into this. But the idea never left. And the state of Ohio, by virtue of the Jobs Ohio program and the Ohio Third Frontier, actually had a program going then. It doesn't have it anymore. It's sort of a pity because it was a good one. But it was called the Innovator Platform Program. There's quite a good amount of funding in the lower single digit of millions, but more than a few $100,000. And the idea that I had that we would use in that previous idea for a company in order to create a machine-enabled system that can investigate and understand cell differentiation, that was baked into a proposal that was submitted back in 2013. And that was submitted through the Case Western Reserve University, even if as I was at the Cleveland Clinic. So sort of a partnership application. And then there were several commercial partners that also participated in it. Now that was actually awarded. And we termed that Ohio Alive. So it was part of the regenerative medicine focus that the state has been focusing on for quite a number of years. And so therefore it started through this program, but I still remained at the clinic, and then was sort of by the state of Ohio put on the path of creating a company. And so that's what happened, because Ohio Alive became Trailhead Biosystems in 2015. And then since then took me on the entrepreneurial journey to build value, growth, and products. Initially, it was built on services, and that wasn't a good idea. So we had to change that path of doing business. And since then, we have changed and focused on making cells for many particular different uses that we can talk about.
Yeah, I definitely want to explore those use cases, the implications of it, the nature of the work you're doing. One question I want to ask, though, it's one of the perennial startup questions, which is why now? But I'm curious, particularly in your context, like what technologies now exist across developmental biology, stem cells, hardware instrumentation, computation? Like what is allowing for you to do effective cell development and in a commercialized, capacity?
I mean, Trailhead is continuously developing an increasing armament of technologies around a core technology that we call high-dimensional design of experiments. Now, this technology is not the only one that exists that can help in addressing this problem of cell differentiation, that's for sure. So it's a good question that you ask. Most of the other technologies that are being developed alongside what we're doing are to a large extent descriptive. The whole field of developmental biology has for decades, its entire existence has been a descriptive way of science. You look at the embryo, you see what's happening, and then you talk about what you see. With the advent of gene editing, the knockout technologies, it was possible to go in and eliminate a single gene or two in a developing mouse, and then you can ask a question, well, what happens if you don't have this component? But aside from that, and that was not human, by the way, that was only in the mouse system or other organisms. So it has been hard over the years to perform experiments where you're at a very large degree change the system, where you change the parameters and look for an outcome. The field of what's called systems biology is essentially stating that this is what you need to do in order to understand biology, because the biology is so intricately wired, with the genes communicating with genes, and as there are thousands of genes, and it didn't come with a blueprint, how can we, going in one gene at a time, ever begin to fully understand this system that we consist of? It's just impossible. So I guess that was the motivation I had, is to build a method that can, in a very powerful way, not just play one string on the violin, but essentially have multiple instruments in play at the same time, so that we can sort of listen to the tune. And this is how high-dimensional perturbation works. You select a couple of instruments that you then play, but you really have no idea what you're going to listen to. But you're going to attempt to listen to things that you know should be important in cell differentiation. So that's what we do. You can't take anybody to just, there has to be a certain understanding of developmental biology before you do these experiments. But together with the machine enablement and what we do, we quite effectively now, I think at least, we quite effectively extract some critical, what we call critical process parameters. And that, by the way, is the term that a process engineer that uses DOE, design of experiments, uses in order to gain process understanding. And so if you want to capture where Trailhead is located, we are located right at the engineering interface to biology. And that is where we employ engineering methodologies, but at a sufficiently high gearing to address biological questions, especially that of control of cell fate. And we are just focusing on getting better and better every day. And since we've been at it for several years now, it's almost becoming a systematic approach for us where we get better for every experiment that we do, because they all speak to each other.
I think it might be helpful to perhaps walk through an example of what this looks like in practice, and maybe diabetes is a good one to work through. But what does this actually look like in practice?
So let's first start with the material that we may start with, right? The stem cells. It's such many people are ethically concerned and so forth, but we're not using any embryonic stem cells here. We're working on induced pluripotent stem cells, which are coming from adult organisms, and they are capable of being reprogrammed, as it's called, through a process where one can overexpress a certain set of gene regulatory factors. When that happens, then they return to a state that's very, very similar to that of the early embryo, and at that point they can expand and grow essentially indefinitely. They also have the capability to become any other cell type of that I just spoke about, all the specialized cells like a hepatocyte or a pancreatic beta cell. Mm-hmm. But that process is one that usually is done only in the context of an embryo. And the embryo is not present here. So how do we do it without the embryo to help? We have to do it in a dish or in a bioreactor. So what we're seeking to do is to guide these pluripotent stem cells one step at a time forward, so it ultimately become the cell we need. And we have to do that just like it would happen in the embryo. So for instance, we call it multi-stage differentiation. The first cell you're going to make is, for instance, if you want to build a pancreas, is an endodermal cell. That's what's called the germ layer from which the pancreas comes from. After you do that, you then generate a field within the endoderm, so you avoid making a liver, avoid making a lung, because that would be alternatives. And then you make the pancreas instead. When you now reach the pancreas, it's like driving to Chicago from here, right? You want to hit Toledo first, hopefully, because if you end up in Pittsburgh, you're in the wrong direction. Right. So we're going to drive to Chicago through the cell stages here, and we're going to go through Indiana at some point. But first, we got to go through Toledo, and then we continue west. So here, the first one was endoderm, and then pancreas. Then we create a pancreatic endocrine cell, and then the endocrine cell has to be a pancreatic insulin producing cell and not some other pancreatic endocrine cell. So you see, at every single step that we employ the technology, we're trying to get further and further specialization to that destination, so we finally end up in the place we want to be at. And this is the same process we need to follow for making a liver or a cell in the brain. It's just a different destination. And that's how to look at it. And if you look at the logo for Trailhead Biosystems, you can see in that logo exactly what I just said. You have like a GPS needle, and that indicates waypoint finding. These are the coordinate sets, but we're moving from a platform to another platform to another platform, and there are 5 platforms in total, and that indicates the cell stage that we move this needle over from state to state. So essentially, the process of HDDOE is the one where we build a protocol up that is hopefully sufficiently good in order to make a good conversion into the cell state that we want.
Okay, that is also very helpful. So to help me understand here some of the implications from a business perspective,
yes,
what is it that you are selling?
So we would like to be actually the leading supplier of specialized human cells, as there is quite a good number of these, and there is also quite a good number of different ways you can use such cells. We're not really in lack of an opportunity or market. So if you think about it, consider these cells as sort of Lego blocks. They come in different colors and they come in different shapes, and they can be used to build different things. So for instance, if you own a 3D bioprinter, and there are several companies that make those, and you want to fill it up with material to print, then some of that material needs to be living cells. If you want to build a skin, you got to have some skin cells to print. If you want to build a liver, you got to have some liver cells to print. And if the ink is dirty, say if the cell is not pure or not functioning, it's almost unimaginable that you can print something that's worth putting into patients or study because it's affected by the poor quality of cells. So therefore, there is a strong need for better cells for, say, 3D printing. But now assume that you, for instance, in the field of, say, you want to make a new drug discovery for Parkinson's disease, okay? And there's a good need for that. How do you do that? Well, you need to perform what's called a drug screen. So you probably have what's called a library of very small molecules, drugs by the thousands, synthesized at a very large cost by some organic chemists or peptide chemists out there. But you need to run the assay where you can see, can you actually find this drug that might be affecting a cell state so you can predict that you can, say, improve in Parkinson's disease. So to do that, you're actually in need for what's called midbrain dopaminergic cells, dopaminergic neurons, because these are the cells that are affected and dying in Parkinson's disease. So as we have built a protocol, a new protocol for these things called A9 midbrain dopaminergic neurons, we then want to offer these to drug discoverers, pharmaceutical companies, to be used in drug screens so that they can get a better hit than any other assay that they could imagine doing. Because if you can't get these cells out of a human brain, it's impossible. And you might be able to do it on some half-cooked mouse cell, but then it's a mouse test. And ideally, you want to do it in a human cell in the right context. And you can even make these cells so that they mimic, say, human Parkinson's disease through genetic editing. So in other words, the cells can be used as tools for 3D printing or for studying human diseases or for drug discovery. And we haven't even yet begun to talk about the therapeutic use. Because the therapeutic use always sits there and people think that that's what you can use the cells for. That what can you do with these dopaminergic neurons? Well, they can likely cure Parkinson's disease. And if they're the right cell, provided at the right amount in the right location in the brain. And there is a company called Blue Rock right now, owned by Bayer, a large pharmaceutical company. And they have gotten approval for a clinical trial of their dopaminergic neurons. And they're recruiting patients right now. I just saw the news report from a Danish-Swedish alliance that have also gotten approval to test. So the therapeutic approaches using stem cell-derived cells is now coming. And it will change medicine. And I think Trailhead's role is to simply develop the best cells and also seek that our cells become tested for therapeutics. And to do that, we're looking to partner. So we would discuss with companies that are interested in developing partnerships for clinical therapy.
Wow, that's extraordinary. The implications, are incredible with that work.
Well, I mean, there's still, just remember, there's not a single therapy based on iPS cells that has been approved. So we're still waiting the first win. And if you look at the diseases that for which, if you have the cells that can possibly be cured using stem cell-derived specialized cells, there are actually quite a number. But the top hitters are, for instance, diabetes, type 1 diabetes. We need pancreatic insulin-producing cells at scale to do that. And that's where the current trial that's the furthest ahead is ran by a company called Vertex in Boston. They bought a company called Semma, also from Boston, and have now gotten the first patients fully off insulin, or close to fully off insulin, as far as I know, in their trial using stem cell-derived insulin-producing cells. So there are multiple companies that are interested in seeing successes in this space. And I hope that, because this is what brought me into this space initially, is the hope that, we can get beyond insulin treatment, but move into curing people instead. Why do we have to inject every single day? Why do we have to be worried? Why do we have to have these complications, all the problems associated, say, living with diabetes for a lifetime, when the cure is so obvious? Just put new cells in. It has been the thing that has always driven me, the simplicity of addressing that is there, but the complexity of making the cell has always been the thing that has followed.
So Trailhead then really revolves around this combination of producing the biological building blocks, the purity of the cellular ink, if you will, the printing ink, and some kind of experimentation around, how that can be used for drug discovery, therapeutics, like you mentioned, not yet, but perhaps in the future, and understanding disease. Is that a fair summary?
That's a fair summary. There's still even further applications of cells, but what we talked about here are by far the most important areas of engagement. And what can you say? It's one of our problems is to find out what direction we should go, not where we could go, because there's so many cells that we can make. The technology that we have is strong enough to likely succeed in making any cell that we set our foot forward to make. But does that mean it's a cell we should make now? So because there's not an unlimited amount of resources available, right? Now we need to focus to create a sustainable business model and grow through that. Understand how to get to a market and understand how these markets operate, because many of them haven't been served at all. And we need to understand what customers and partners are looking for in the cells that we deliver without messing up too much along that way. And so therefore it's a constant sort of testing forward, getting out there, asking and meeting people. Cleveland is not Boston. It is not San Francisco, and it's not San Diego. So we are not in the wheelhouse of or ecosystem of biotech in general. So most of what I'm talking about here has to happen both nationally and internationally outside that of Cleveland. And that's just one of the challenges we have to deal with.
I know you have raised around $10 million towards this effort. Maybe just kind of give us an overview of what the company looks like today and where your traction and focus is as you are trying to parse where should you go versus where could you go.
I think so. What we figured out is that where we should go is make some cells and sell these and get a revenue basis in this space, because it's entirely doable. Then all the therapy comes automatically on that basis, because the cells are there. They're not going to go away. And we'll find paths for that. So we are focused on production. And we have raised into creating a better set of capabilities into making the cells, growing the cells, scaling them, and hiring in on sales. And then we've decided to stay here in Cleveland. We were able to purchase a facility that we're going to move into actually in not more than a couple of weeks.
Oh, wow.
It's located in Beechwood. We have fitted out a reasonably well-sized lab space, tissue culture facility, some manufacturing area, bioinformatics, employee lounge and offices out there, so that we can grow in our own space. And I'm just looking forward to that, because it'll be a new phase of the company, and it's a little bit overdue, I would say.
Oh, that's very exciting. What is like the next thing that you have to validate as part of this journey? And I guess a different way to ask that is, in retrospect, assuming you achieve success, I guess what does success look like, and what are the steps along that journey?
I mean, most people think about success as being some point in the future where, say, a revenue milestone has been achieved, or an acquisition, or an IPO, and so on. They're all sort of meaningful demarcations along the way. But to me, what I see when I go to work every day with the team that we have here, and how much that's happening, how much people are growing, and the type of understanding that we get just from our activities, that's success. The labs in the lab, and the just seeing a company being built is success. Yes, there are some other defined milestones that we're going to put up, and we will, and we have to, because that's how you have to work in a startup company, because those deliverables are key. But in and of themselves, they shouldn't be the success milestones, because there's a lot of things that are unexpected, and it happens, and that can, if not more, impact the success criteria. And funnily, as a scientist, I have to admit that science never moves in sort of the fully predicted way, right? Usually there are some events that just stand out as being transformative. I think for us, the first transformative event was when we did the first large experiment and we could actually see. And that's many years ago now, but that was, at that point, I just knew. So when you combine value building and science, it gets risky, because science can take forever. Scientists think that they can be at it forever. Companies have a limited amount of time to focus on creating particular milestones are of commercial interest. So how do you create the pickup of the value of a scientific process? I don't have the full answer to that, except to say that I'm believing that we have to gear up that science to really deliver, and then we keep the scientists extremely focused so that they don't get too much into another rabbit hole, and lose that focus, which is all too easy. We have a little bit of a motto here, actually, and from a reflection standpoint, so it's we change the scientific process. And that's a big statement to state that, because you can't change the scientific process, right? It just is what it is. But if I try to explain why I'm saying something like that, it's because all the scientists that we're essentially training, teaching them how to get grants, teaching them how to do experiments, is all based on hypothesis-driven research. And hypothesis-driven research is by definition then rooted in the human mind of a hypothesis. You got to come up with an idea. And then by virtue of your training, you will test this idea to either prove it or show that it is false. And that can take a good amount of time to do, and you buy into this idea of yours. So you might get cognitively biased towards what you want to see. But the biggest problem is that it's a human-centric process. I call that the anthropocentric problem here. Anthropocentrism essentially just means that we tend to view ourselves as superior as a species. And you can see that in so many different things in society, where we're the apex species and so forth, right? But in science, it also means that we tend to think that it takes the Einstein to understand the world we live in, because it's the most intelligent mind that can do that. But to me, I mean, the biology world that we're entering into here is so highly dimensional that the human brain is incapable of assembling that. It cannot be done. So in other words, what can we do to sort of take the human out of this process and let the machines help us, so that we will amplify the experimental interrogation? In other words, try to avoid thinking about all the hypotheses. Don't do that. Just test your way through. Get the response back and listen to what nature just told you. And this is sort of that, the other perspective to this is to see, aren't you yourself the biggest roadblock? And I think that's what dawned on me back in 2011, that I was beginning to realize, despite all the years I've spent as a developmental biologist, that I simply wasn't capable of doing this. No matter how much I would spend time, years, hiring people, publishing papers, we were not going to get through this problem. And still with whatever we have, what we're applying here, these are sophisticated problematics that we're going through, but it's a lot better than what it was before, when you could test ideas one at a time. So yeah, changing the scientific process just means that we're sort of trying to make ourselves a little bit smaller, help it with the machines. One example I can say is that everybody can sort of realize or relate to is take two people, give them a task, it's the same task, dig a hole, okay? You can go find the strongest person on earth you can find, Schwarzenegger style, okay?
Sure.
And you go dig your hole over to the left, and then I'll go find the little weakling over in the other side, and then I give him the excavator. Okay, and then we'll come back tomorrow and see who was digging the biggest hole. It's the same thing, right? It doesn't take physical strength. It doesn't take mental strength. It takes the enablement. And that's where I think science needs to go. And I think most of the technologies that you're talking about that are involved here, they are all helping us do more as scientists. But I'm mainly focused on what's called the empiricist side, the experimentalist side, those that actually do the experiment, not those that gather a lot of data and try to make sense of what they looked at. You can compare to the physicist. You can have a theoretical physicist, or you can have a physicist at CERN that runs a particle accelerator. I like to be the person with the accelerator, not the one only theorizing, because eventually you do need the data to understand whether or not your theory was correct or not.
No, I think it makes a lot of sense. Building a company is one of the more effective ways to test the efficacy of those ideas. How optimistic are you that, and in what time scale, that some of these challenges that humans face biologically could be solved?
That's a question you should never ask a scientist, because if the scientist comes back with the time frame, he's completely bonkers. So I've always learned never ever to put years on anything when it comes to when do we have the cure. And so many years ago, I was hoping that the cure for diabetes would happen at the century of the discovery of insulin. And that would have been last year, because insulin was discovered in 1921. So that didn't happen. So we're still a couple of years behind, but I was hoping that it could have happened. But I think the proof of concept has been shown. It's just not approved as a medical therapy yet. So it will come, but I'm not the one to say when. I just don't dare. Because most of these, from a therapeutic perspective, as you're very aware, takes years of regulatory fighting to get and testing to get new therapies approved. But they will come. There's no question about it. They will absolutely be here. But when they will be here, whether it's a decade out, maybe even before for some, or whether we have to wait 2 decades. And what I can say is that the process of development now is exponentially accelerating, right? So what was happening over 1 year, say 10 years ago, probably now happens within a month or so. I'm speculating there. I can't say for sure. It also depends on what you're looking at. But it's very clear that we can move faster now than we ever could. And I expect that acceleration to continue. I don't know if there is a Moore's law on the scientific side, but it's the same principle.
Yeah. What has surprised you most about your journey so far?
I don't really know. You got me on that one. let me think a little bit about that.
Sure. Yeah.
Yeah, I think what has surprised me is that, usually the worst outcome that you think in a given situation later can turn out to be one of the best. And you had no idea that, that is the failed attempt to make a diabetes company that led to Trailhead. It was wonderful the first one failed, because it would never have had the opportunity that this one has, I think. I'm very happy that I don't have the perfect eyesight, because had I been a fighter pilot as I wanted to, I would have been ferrying some people in an Airbus or something like that today, or whatever, right? I mean, you just don't know. So these events in life that you think are setting you back are more often than not just throwing you forward. You just don't know it. But you have to get forward, and then you begin to reflect on, and then so I've just learned to accept fate. So if there's something that doesn't go my way right now, I say, well, maybe it's not meant to be. So we'll see what then comes. We can do the best in the moment that we are, and put our integrity forward, trust in our ability to change, and then lean on others, right? I mean, Trailhead is not me. I have to say that it's the idea perhaps, but it's the team. I mean, honestly, I'm not doing this science. You have to interview the folks that are actually doing the experiments that are in our company now. So, and if Trailhead becomes successful, it's also not because of me. It's because of the team, 100%. And I'm totally in awe of how such a collective can form, and that's why I love the word company. It's the right word. Where I come from, we don't call it that when you build a company. We call it something different when directly translated. But the word company is a great name because it's a company of people. And as much as we talk about technology here, it's absolutely a dead thing. Technology doesn't do anything. It is when the humans come in and use it that there is an impact. And the value of a company lies in its employees. That then can be amplified with technology, but it's always the employees. And I'll just call it the team. So there you go, going back to the success thing. If you're able to go to work and you surround yourself with unbelievably gifted individuals, that's success.
I love that. That's awesome.
To me, at least.
Well, we'll tie into our closing question here, which we ask everyone on the show, which is completely unrelated to the work you're doing, but it's for your favorite hidden gem in Cleveland.
Yeah. So one can think about, it's a walk in the park out in metro parks, is this the place to go for the best sandwich or stuff? But I don't know. I actually think the hidden gem in Cleveland is Cleveland. To me, it was off the radar. It was flyover territory in diabetes. I think we've been successful here because we're in Cleveland sort of hidden. I don't want to be condescending about this, but we don't really, it's very difficult to think about, a large number of biotechs in Cleveland. And I think it's been good for this little company here because you see that sometimes, they're venture capitalists. They want to start a company and set aside funds, and then they found a crackerjack team, and then they say they're all in stealth because they don't want to let anybody know what the heck they're doing up until the point when they come out of stealth, and then the world can see how much of a great idea that they've been now working on collectively for a year. I mean, Trailhead has never been in stealth. But we've been in Cleveland, and it's been working fine because we got the time, we got the support. There's a lot of buy-in in the state of Ohio to what we're doing, and I've just been, one can say that the area that we're in fits in what is referred to as deep technology, and it just takes time to learn this stuff. Right, we're doing things in a new way. You're going to make a lot of mistakes, but you learn. But you need time. And I think we got time. We got early support. And now we just need to prove that we can make an impact. And that impact is by delivering material. It's not our technology, because the technology doesn't matter to anybody unless we can provide cells. So And if so, there's a market strategy, there is a production strategy. There will be a lot of learned principles along that we have to go through. I think we can solve that, I hope. But so there you go. I mean, the transitions are natural, and Cleveland, to me, it grows on you. I mean, raising a family in Cleveland is not a bad place to raise a family. That's why we're still here. I think that Cleveland has a great opportunity to attract other biotechs out of Boston, out of San Diego, into Cleveland. It's not just the cost of living. It's not just the cost of lab space. It's a good place to do it. Most of the employees we get here, we can find in Cleveland.
Mm-hmm.
Some on the more senior side, we have to look out of state for, just because it is. And there is a general excitement and also a very down-to-earth attitude to how to get stuff done here. One of the things that strikes me as important is, so I've been talking to several people in the Silicon Valley, and the entrepreneurial mindset is flowing through the veins of everybody out there.
Yeah.
And it can take you into the air, and all of these could be's are then, and you get dizzy. You get really dizzy. So what is all of this? Is it all make believe and so forth, right? And most of it is by and large just speculation. And for sure, they showed the world how to build technology companies. But the mindset itself is just causing confusion. I mean, to me, it causes confusion. Coming back to Cleveland, you're grounded again. Here you're grounded. You can focus, and that's what is also something that I think is important, that we can very, very quickly get distracted, and we don't necessarily need all the noise. But now we need to make some noise. Now we need to get out and talk about what we can do. So we have to turn it around a little bit.
No, I love that. Cleveland as the meta hidden gem of Cleveland.
I think many Clevelanders would probably think like that, because we all know the statements of mistake on the lake and whatever. but then again, people still stay, and many are loving coming back. So I like to see Cleveland in 10, 20 years, hopefully continue along the trajectory that it's put itself on. There's still a lot of work to do, I would say. I'm not fond of letting urban blight stand, and let's just face it, we have that. Where I come from in Europe, such is just not tolerated, and it'll be fixed. But then I can say what I've seen over the last 10, 15 years has been remarkable. And I just hope we can continue to do that. But we have to do it together, and we have to trust each other.
Well, yeah, and I really appreciate you coming on, for sharing your story. Your perspective is fascinating, inspiring work. So thank you very much.
Thank you so much, Jeffrey. Such a pleasure being here.
If folks had anything they wanted to follow up with you about, what would be the best way for them to do so?
I mean, they can write to me. That's probably the best. We also have a on the website for Trail Bio. www.trailbio.com. You can contact us. I'll be happy to take your listeners' comments and see if I can get around. I can't promise to reply because the days are quite full, but yeah.
Awesome. Well, thank you, Jan.
Thank you.
That's all for this week. Thank you for listening. We'd love to hear your thoughts on today's show. So if you have any feedback, please send over an email to jeffrey@layoftheland.fm, or find us on Twitter @podlayoftheland or @sternhefe, JEFE. If you or someone you know would make a good guest for our show, please reach out as well and let us know. And if you enjoy the podcast, please subscribe and leave a review on iTunes or on your preferred podcast player. Your support goes a long way to help us spread the word and continue to bring the Cleveland founders and builders we love having on the show. We'll be back here next week at the same time to map more of the land. The Lay of the Land podcast was developed in collaboration with the Up Company, LLC. At the time of this recording, unless otherwise indicated, we do not own equity or other financial interests in the company which appear on this show. All opinions expressed by podcast participants are solely their own and do not reflect the opinions of any entity which employs us. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. Thank you for listening, and we'll talk to you next week.